Hey everyone, welcome back again. This is presentation number 4, and today we are going to look at the rest of the story. Well, at least as far as molecules are concerned. Now that we've disposed of 70% of the molecules that make up the average organism, now it's time to turn our attention to the remaining 30%. Those molecules that are built on a backbone of carbon. So in this presentation, we're going to discuss, first of all, why carbon? What is it that's so unique about a carbon atom that allows it to do this? And why can't something else that has similar chemical properties to carbon do the same thing? Then we'll talk about the nature of organic molecules and just how diverse organic molecules can actually get. And once we talk about that diversity, we then have to start thinking in terms of the large molecules that form cells. They will be the subject of our discussion in the next presentation for chapter 5. But we start getting into those large molecules, or start thinking about those large molecules by thinking about how a basic carbon skeleton can be modified to create the multitude of different types of organic molecules that actually make up cells. And that we'll get to right at the very end of the chapter. This is not a terribly long presentation compared to some of the others that we encounter along the way. And in fact, it's not as long as most of the presentations that we've already covered in terms of the number of slides. However, some of the ideas that we do need to discuss here and some of the background information and additional information that I want to give you will probably expand this presentation out to about an hour and a half to 2 hours, as is roughly the size of most of the presentations this semester, unless we get into a really big one. And that's because there's a lot of interesting things that we can talk about here. And one of the things that I want you to realize is that while the textbook and while the PowerPoints give you the basic information that you need to know, the more you can learn about a topic, the more detailed information, the more interesting information, the more relevant information, the more connected information you can take on board that makes the ideas more personal, the more likely it is that, number one, you'll be able to learn the material to the level of mastery, and number two, you'll be able to retain it, which is, after all, what our goal should be this semester. And one of the things that I would always challenge you to do as you begin every chapter, as you begin every learning experience, is to ask yourself, what is it that I want to get out of this? What am I really learning this material for? Because if you haven't asked that question, it probably will become challenging to try and understand the material if you have no real concept of why you're doing it. So ask yourself that question every time you sit down to start to learn the material. So, having given you a little bit of a lead in, let's start talking about carbon. So the first thing that we recognize when we start thinking about life and we start thinking about life from the chemical perspective is, as I said in my introduction, that once we dispose of water, the vast majority of everything else that's left is built around carbon, and specifically chains of carbon atoms linked together with other things attached to that chain. And while that may seem like a relatively simple idea to grasp, the diversity of molecules that carbon can build simply by linking together in chains is overwhelming. What you may not realize, but what is very apparent to anyone who pursues a degree in chemistry and a career in chemistry, is that all of our industrial chemistry, that huge complex industry that represents our industrial chemistry, is built around organic chemistry, carbon-containing molecules. And there are lodged at patent offices around the world millions upon millions of patents for unique types of organic molecules that have been developed by humans over the course of the last 150 odd years that we have been able to create synthetically organic molecules. And therein hangs part of the tale for this chapter, for this presentation, because up until the late 1820s, it was always thought that carbon-containing molecules could only be made by organisms. That's why these were referred to as organic molecules. And the study of those molecules is referred to and was referred to initially as organic chemistry. These molecules were thought to be only possible due to the chemistry of organisms, that it was simply impossible to create these synthetically. That would change at the end of the 1820s, as you'll see. But up until that point, a philosophy called vitalism, which basically said that there were special properties associated with living material that was only found in living material and could only be associated with living material, and that human beings were incapable of doing anything in terms of chemistry, at least, that organisms could do. And that organisms, that life spontaneously emerged out of organic matter. That organic matter gave rise to organisms, and that this was something that occurred spontaneously. This was all encapsulated in this philosophy of vitalism. Vitalism would suffer some injury as a philosophy in the 1700s and particularly in the late 1700s, which would set up the observations of one man in the late 1820s that would basically turn the whole notion of organic chemistry on its head. And then the final nail in its coffin would be driven home when Charles Darwin published On the Origin of Species in 1859. That finally drove or closed the lid on the whole notion of vitalism as an explanation of life and its origins. But it would all begin with a simple chemical observation made by one man in the late 1820s. So we have this notion of organic molecules being the product of organisms. That would change, and that would give rise to our vast industrial chemistry complex that we have around us today. And when you think about all the things that our industrial chemistry can provide, synthetic fabrics, plastics, drugs, all sorts of chemicals, all of those are the product of our ability to create organic molecules. And even when we think in terms of the observation that most of the drugs that we use today, or most of the common drugs that we have used in the past, up until probably the 1980s, 1990s, were usually derivatives of chemicals that were found naturally occurring in nature. A lot of our drugs actually trace their origin back to naturally occurring compounds in plants and bacteria and fungi, all sorts of origins for those chemicals. And subsequently, we learned how to create them synthetically so that we don't have to rely on the organisms as sources of those chemicals anymore. But they all have their origin in organisms. But fortunately, our industrial chemistry, our understanding of organic chemistry, our knowledge of the reactions of organic chemistry now allows us to synthesize molecules. And we can take it one step further when it comes to the pharmaceutical industry, at least. We can design chemicals, we would call them drugs, but they're still chemicals, specifically to target a particular biochemical system in an organism. So to target an enzyme or to target a receptor protein, because we understand the nature of the structure of the molecule that normally binds. We talked about that back in chapter 2, in the previous presentation, or in the chapter 2 presentation. just simply understanding the structure of the naturally occurring molecule and our knowledge of organic chemistry and our ability to carry out syntheses in the laboratory allows us to create designer chemicals, designer drugs. All of this traces its origin back to the observations of the one individual that I keep referring to without naming him, but we'll get there. And the simple observation that you can, in fact, synthesize naturally occurring organic molecules in the laboratory without life being present. And that would be the starting point for everything that we know today about our modern industrial chemical complex. Kind of important idea. But just to finish off this section before we start talking about why carbon, when we start thinking in terms of the diversity of life, one of the things that we will see in the next chapter is that while there are only a limited number of types of large organic molecules, four in fact, that we recognize, there is a huge diversity of these molecules, and typically every type of organism has its own unique suite of these very large molecules. And it's that diversity of molecules, it's the diversity of organic molecules, even though we can divide them up into four separate classes. Within those classes, there's a huge diversity of molecules that fall into each of those classes, and different organisms make different combinations of these diverse molecules. It's that diversity that accounts for the diversity of life, if no more than at the DNA level, but we can expand that to all the different types of large organic molecules that we'll be discussing in the next chapter. So having an understanding of the role of carbon, why carbon, having an understanding of the diversity of shapes and types of molecules that carbon can form, and then how those basic shapes and structures can be modified by adding things to the basic skeleton, allows us to account for the diversity of chemicals and therefore the diversity of life. You can see how one leads to the next. It's a series of connected ideas that allow us to reach the ultimate solution to the question, why is life so diverse? We start with simply understanding what it is that's unique about carbon, why it can what it can do, make those diverse types of molecules, how those can be modified to make the diverse types of molecules that are found in cells, and how that leads to the diversity of organisms. That's the daisy chain. We need to begin with understanding why it's carbon and not something else. So the main reason why it's carbon, or the starting point, is the simple observation that carbon can form four covalent bonds. We talked about that in chapter 2. The idea that when carbon rearranges the S and P orbitals in the second energy shell, that is its valence shell, into those four hybrid molecular orbitals, there is now the possibility of forming four covalent bonds. If those four covalent bonds are formed with hydrogens, we get our old friend methane, which we talked about back in chapter 2. Remember, it is a symmetrical molecule, and you can see the four hybrid orbitals linking up to the hydrogens. And remember, each of these molecular orbitals, these hybrid orbitals, contains a pair of shared electrons, the basis for the covalent bond, one coming from the hydrogen, its only electron, and one electron coming from the valence shell of carbon. And it's this shared pair of electrons forming the covalent bond that satisfies the hydrogen's requirement for a full valence shell and partially satisfies the carbon's requirement for a full valence shell. To finish out the carbon, you need four covalent bonds, four shared pairs of electrons, to fully fill the carbon's valence shell and satisfy the requirement of a full valence shell, 8 electrons, the octet rule. And so this is the beginning of our understanding of organic molecules, just this simple observation that carbon forms four covalent bonds. And in fact, methane is considered to be the simplest of all organic molecules. It's the smallest and the simplest, and it represents the starting point from which we start to think about all other organic molecules. But we still haven't answered the question, why carbon? Why is it that carbon forming these 4 covalent bonds can do what it does and no other atom can do this? Well, if you turn to the periodic table in the textbook, and I'm not going to jump to that because I don't have a picture of the periodic table here, but if you go and take a look in chapter 2 and you take a look at the periodic table in chapter 2, you should recall that any element that, or elements that sit in the same column, have the same number of electrons in the valence shell. Remember, the row tells you which valence shell is being filled. The column tells you how many electrons are in that valence shell and how much room there is to fill out that valence shell, how many electrons are necessary to fill out that valence shell. And carbon sits at the top of column number 4 in the second row. So its valence shell is shell number 2, and it has 4 electrons in that valence shell because of its location. So any other element that sits in column number 4 should also have 4 electrons in its valence shell and therefore be capable of at least forming 4 covalent bonds. So as soon as we look underneath carbon, we find the element silicon, given the symbol Si. So we fire up the pen. So silicon. Symbol Si also has 4 electrons in its valence shell. So arguably, it ought to be able to form 4 covalent bonds, just like carbon, and it does. So the question then becomes, if silicon can do this, why doesn't silicon form the basis for life, or potentially form the basis for life? What is it about carbon that means it can do it, but silicon can't? Well, the answer lies in comparing a covalent bond between 2 silicon atoms versus a covalent bond between 2 carbon atoms. And when we do this comparison, it's also important to understand how stable these covalent bonds are in the presence of oxygen, molecular oxygen, O2, oxygen gas, because it's that really tells us why it can only be carbon under the circumstances that exist here on our planet and not silicon or any other element. Because it turns out that the most or the largest reserve of silicon on our planet is silicon dioxide. So let me put the note here. That should probably be a capital E, I guess. SiO2. The largest reserve of carbon is, the organisms and their fossilized remains so that is life, past, and present. And therein lies the reason why it's carbon and only carbon and not silicon. If you had a silicon-based life form, it could only survive in the absence of oxygen, which, if we think about the history of our planet, for about the first 2 to 2.5 billion years, that might have been possible because there was very little oxygen in terms of free oxygen present on our planet. It would only be with the evolution of oxygenic photosynthesis, the sort of photosynthesis that plants do, but in the form of simple photosynthetic bacteria called cyanobacteria, that oxygen would start to accumulate first in the ocean, and it would combine with iron to rust the iron, and that would precipitate into the large deposits of rusty iron that we mine today to make iron and steel. And once all of that iron had been rusted, combined with oxygen, then the oxygen would start to accumulate in water. But because oxygen doesn't dissolve particularly well in water, it started to accumulate in the air pretty quickly. So it was life itself that started the accumulation of oxygen through photosynthesis. So that by the time we get to around 2 to 2.5 billion years ago, we start to see substantial amounts of oxygen in the atmosphere, and any combination of silicon atoms that might have linked together in a chain would have collapsed in the presence of oxygen, because oxygen reacts very easily with a silicon covalent bond and breaks it to form silicon dioxide. What is silicon dioxide? Well, that's sand, common garden variety sand. When we melt it and allow it to set up again, we get glass. So your glass window is actually silicon dioxide. And so silicon or the silicon-silicon covalent bond is not particularly stable in the presence of oxygen. Neither, for that matter, is a carbon-carbon covalent bond. And you can test this by taking a little bit of cooking oil, putting it in a glass and just leaving it out on the kitchen bench for a little while. And you'll notice that the oil starts to go from a clear color or a light yellow color to something that is darker and browner, and it'll start to take on a funky smell. And the term we use for that is to say that it's going rancid. But what really is happening is that the oxygen in the atmosphere is attacking the covalent bonds in the fat and starting to break them down. But therein lies part of the tale. That fat, under normal circumstances, would be part of an organism, a plant or an animal, depending on where the fat came from. And while it's inside the organism, it's protected because organisms have ways of dealing with the oxygen. That's why we are able to live in an oxygen-containing atmosphere. And in fact, life went one step further, not only figured out how to deal with the oxygen, but to actually take advantage of oxygen's reactivity and use it to life's advantage by being able to create boatloads of energy in the form of ATP. That's what we'll learn about when we do chapter 9 and we think about cellular respiration. That's life's ultimate interaction with oxygen, taking advantage of oxygen's reactivity and using it to life's advantage to make the necessary energy to support organisms through cellular respiration. Again, a very old process tracing its origin back to bacteria and the evolution of oxygen through photosynthesis, but nonetheless advantageous. And it allows life not only to deal with the oxygen and its reactivity, but to take advantage of it and thrive in the presence of oxygen. So as far as our planet is concerned and the presence of our oxygen-containing atmosphere, about 20% oxygen, there is no other element that can do this. It has to be carbon. Silicon simply couldn't do it. What adds to this is the observation that even in an oxygen-free atmosphere or an oxygen-free environment, silicon can only form chains of about 8 atoms long. When it comes to carbon, we don't know the limit. Carbon chains can be of very long lengths. The longest ones that are found routinely in organisms are around 20 carbons long in the form of fats. But in our industrial chemistry, we can create inordinately long chains. There's virtually no limit to the length of chains that we can form by linking carbons together through covalent bonds. And that again reinforces the fact that under the circumstances that we find here on Earth, the chemical circumstances here on Earth, it has to be carbon and only carbon. Could silicon or other elements form a basis for life elsewhere? Yeah, of course. As one of my science heroes, Albert Einstein, not a bad hero to have in science, said, the universe is not only weird, it's weirder than you can imagine. And as much as we may not be able to imagine a situation where a silicon life form would exist, given that there are so many possibilities out there, there is likely to be somewhere, chemically, a silicon-based life form. It may be very simple, but nonetheless, it might exist. And certainly the writers of the original Star Trek series thought that that was the case, because there is an episode. I am of the generation of the original Star Trek. Yes, I am that old. That they wrote into one of their series a silicon-based life form. They kind of destroyed the image, however, for anyone who understood the science by having Kirk and Spock standing next to it so Spock could do one of his Vulcan mind melds to try and figure out what this thing was thinking. And of course, as we know, Kirk and Spock are oxygen-breathing organisms. They survive in an oxygen containing atmosphere. This silicon-based life form probably could not have survived in the same neighborhood as they were in. But nonetheless, it didn't escape the notice of the writers of Star Trek that the possibility of a silicon-based life form was certainly there based on the chemistry. And that's not an unreasonable hypothesis, given that the universe probably has many, many different types of life forms. Yes, I do believe that there is life elsewhere in the universe. What it looks like, I have no idea. Probably not like us, though. But that's a story for another day, I suspect. Let's move on. So now that we've established why it's carbon and only carbon, and can't be anything else, at least in the chemical conditions that we have here on our planet, what's the upshot of that? What do we know is the possibilities then as far as organic molecules are concerned? Well, as I said in the lead up and the introduction to the presentation, it would take the observation of one man in the late 1820s to change the whole notion of what an organic molecule actually is. So I talked a little bit about vitalism, and that was the reigning philosophy at the time that Friedrich Wöhler, a German chemist, accidentally created urea. Now, Wöhler was in an ideal position to realize that he had made urea because he was studying the chemical compounds that were found in urea. He would take urea from organisms, and I'm sure he probably made his own contribution to his research. And he would place the urea in a beaker and allow the water to evaporate, and then take a look at what was left behind after the water had evaporated. And one of the substances that he recognized were yellow crystals of urea. It is a substance that is filtered from the blood by the kidneys. It's a way for the body to eliminate nitrogen-containing waste from amino acids found in proteins. And so if you eat a lot of protein, you need to drink a lot of water to make sure that the kidneys have enough water to be able to eliminate the urea. Otherwise, you're asking your kidneys to work really hard and you can actually damage your kidneys under those circumstances. So urea is a common component of urine, and it's there because it's the nitrogen-containing waste that is secreted as a result of the breakdown of amino acids if you use amino acids as a source of energy, which you can do. And so Waller was really familiar with the look of urea. And one day when he came into the lab in 1828, he noticed that there was some of these yellow crystals and presumably urea sitting in a beaker. The problem was he knew that he'd not had urine in that beaker. In fact, he'd had some inorganic chemicals, not organic chemicals, sitting in that beaker. He had just forgotten to wash it out. And lo and behold, suddenly there appeared in this beaker urea, or crystals that looked like urea. And so he did his chemical tests that he knew would identify the crystals as urea, and sure enough, that's what they were. Now, of course, at this point, he's going, that can't be correct. I must have made a mistake. I must have had urine in there. I just can't remember having it in there. So he set out to do this deliberately and took the chemicals that he suspected he'd had in there, or that he knew he'd had in there, got a clean beaker, made sure it was washed out, that there was no possibility that there was urine in there, put the chemicals in there, put it aside, and waited. And sure enough, in a short period of time, a couple of days or so, there were the crystals of urea in there. This was not an accident. This was actually something that was happening. By mixing the right chemicals together, he could create urea, an identifiable organic molecule, the product of an organism, by mixing together chemicals that were most definitely inorganic, not the product of organisms. And you can imagine Waller's surprise at being able to do this. Because here was the possibility of being able to make an organic molecule in the absence of life, something that flew in the face of everything that was accepted about organic molecules up to that point, that they simply could not be synthesized artificially, that they could only be made by organisms. And Waller's going, wait a minute, that's not true anymore. I can make urea in the absence of organisms by simply mixing the right chemicals together. And ultimately, Waller would experiment with how to make urea, and he would come up with a more effective way of doing it, which would become known as the Waller synthesis for urea. And we still use it today because urea is a common component of artificial fertilizers. And why do we have artificial fertilizers? Because we can synthetically make the organic nutrients that plants can utilize from the soil. They do exist naturally in the soil as a product of bacteria and other microorganisms in the soil. But if we want to enrich the soil and add to the soil, or we want to supplement what's already in the soil, we can add artificial fertilizers, and many of the components of those artificial fertilizers are things that we can synthesize, including the urea that we have synthesized artificially in chemical factories. And so it was this simple observation of Waller's, this ability to be able to make urea in the absence of organisms by simply mixing together the right combination of chemicals, that turned the whole notion of organic chemistry on its head and opened the door for chemists to start trying to make all kinds of different organic molecules, ones that they already knew from life, but also to start imagining all sorts of new types of organic molecules that didn't exist in organisms, but might have some uses for other purposes. And that was the beginning of our industrial organic chemistry. And perhaps one of the most famous chemical companies on the planet that is still around today in, not in its original form, but it is still around today. One of the biggest chemical companies on the planet, Bayer Chemical, was built on the back of aspirin, because aspirin is a derivative of a substance made by plants called salicylic acid. And in traditional medicine, particularly in Europe, if you had a toothache, the healer, the shaman, the medicine man, would give you a piece of willow bark to chew on, the inner bark of the willow tree, which contains a lot of salicylic acid, and it would act as a painkiller. And the chemists at Bayer were able to isolate salicylic acid as the active ingredient and then figured out a way to make it synthetically. The problem with salicylic acid, however, is that it irritates the stomach really badly and causes the stomach to bleed and gives you all sorts of stomach aches as a consequence. So they looked at the basic shape of the salicylic acid molecule and figured out a way to modify it a little bit so it was less irritating. And that became aspirin. And that was the first commercially available painkiller. And out of that would come all the subsequent painkillers that belong to a family of painkillers called NSAIDs, non-steroidal anti-inflammatory drugs, or NSAIDs. And that includes aspirin, Aleve, Tylenol, ibuprofen and acetaminophen. I forget which is which. One is Advil, one is Tylenol. But these are all derivatives of the original salicylic acid / aspirin molecule, and their modifications are designed to make them less irritating to the stomach. Not always necessarily successful, but they, in terms of reducing the irritation to zero, but at least reducing the irritation to the point where your stomach is not bleeding every time you take a painkiller. But it all started with Walden. The simple observation that you could make urea, a substance up till then only known to be the product of organisms, by mixing the right chemicals together. And so begins our industrial chemistry. Quite amazing to think about it when you understand the story, that it was just a simple accidental observation, a mistake, an honest mistake, forgetting to wash a beaker. But that's the nature of science. Sometimes some of the most amazing discoveries, some of the most fundamental discoveries are made by making a mistake, forgetting to do something, or doing something incorrectly, and realizing that what you've done has actually led to a result that otherwise you might never have seen without making the mistake. Anyway, we need to move on and keep talking about carbon. So let's move on and start talking about the flexibility of carbon. We know that it has to be carbon. We know that it can't be anything else other than carbon. Waller's now indicated that it is possible to synthesize organic molecules in the absence of life. The ultimate expression of this would come after Darwin published On the Origin of Species, and people started to realize that natural selection and evolution represented the best explanation for the origin of the diversity of life. That in fact, descent with modification explained why organisms changed over time, and that organisms that shared common features did so because they were descended from a common ancestor, and the differences between them were a product of tinkering and modification of a basic structure to be put to different uses depending on what the different organisms were using them for, which meant you should be able to trace back in time, ultimately, if you go far enough backwards, to the ultimate ancestral cell that is the ancestor to all of life as we know it. But then if you take that one step further backwards and go back to the time before cellular life, to when there was no life, could you find conditions that would allow for the synthesis of the molecules that we know make up cells and are fundamental to cellular life as a product of a non-cellular based chemistry in the environment? And that would be first proposed in a hypothesis in 1921, which would become known as the Oparin-Haldane hypothesis after the two biologists, one Russian, Oparin, and one British, Haldane, who first proposed independently this same idea, that the chemical conditions on the pre-life or the pre-cellular life Earth were such that it would allow for the synthesis of the simple organic molecules that could be assembled into the more complex organic molecules that in combination and in support of each other would eventually assemble into complexes that would lead to the appearance of the first simple cells. But it would all have to start with the ability to be able to create these simple organic molecules in an abiotic, in other words, non-life situation. That hypothesis would be put to the test by an American graduate student named Stanley Miller, who in combination with his supervising professor, Harold Urey, designed a device that looks something like this. They did this work at the University of Chicago, and in 1956, Miller published the results of his early experiments using this device. So Oparin and Haldane hypothesized that, number one, the Earth would be really hot because it condensed out of material left over from the formation of the sun, so rock and dust particles, things like that. And we know that the Earth still contains a lot of residual heat in its inner core. We see that in the form of the molten rock, the lava that comes up in volcanoes and things like that. So early on, the Earth was probably really hot with a thin crust of solid material over the top. Any water that existed was probably not in its liquid state, it was probably more in its gaseous state. So Miller designed the experiment to contain gaseous water by boiling water in a flask and allowing the vapor to enter the experimental chamber. In that chamber, Miller placed the gases methane, ammonia, and hydrogen. Why? Because Haldane and Oparin considered that these were the gases that likely formed the Earth's simple atmosphere early on, because these gases are present not only in our solar system, but everywhere in the universe in large amounts. In fact, hydrogen accounts for about 98% of all the atoms present in the universe. Yeah, a single element accounts for the vast majority of the matter that we can see in the universe. The other 2% is made up mostly of helium, and then a tiny little fraction is everything else. It's quite remarkable, that. So all of this is in gas phase because, again, it's part of the gaseous atmosphere of the early Earth. Then Miller also allowed for electrical activity, sparks in the atmosphere. Why? Because if you look at any YouTube video of a volcanic eruption, and you can find plenty of them on YouTube, as you watch the ash clouds get ejected into the air, forced out of the volcano and ejected into the air and many miles up into the atmosphere, you will see periodically major lightning flashes inside the column of ash and also flashing outside of the column of ash to the ground. And because the Earth was very hot, volcanic activity was probably very widespread, and that would mean that there would be the possibility of lightning discharges, electrical discharges in the atmosphere because of all the volcanic activity. That was the hypothesis. So hence the electrical sparks. So Miller sets up this device and starts it percolating. And after whatever happens in the reaction flask takes place, he collects the gases and condenses it out, or condenses the water back. And whatever's been produced in here, of course, gets captured in the water and condenses out in the water. And the water gets recycled back into the flask where it's boiled. Anything that's not water, of course, gets retained in here. And over time, it was very obvious that something was happening, because the water solution that was being condensed was changing from clear to something that was light yellow and eventually would turn into an orange-brown sludge. Something had happened. Something was being produced. And so Miller would tap into the solution and collect whatever was being produced and then do a chemical analysis to find out what was being produced. And what he discovered was that he was able to make a couple of the common amino acids that are found in proteins in organisms, a simple sugar, and ultimately playing with this device, because this device would become part of Miller's lifelong work. He would use devices like this and modifications to these devices for the rest of his life, because he became a great proponent of the idea that the chemistry of the primitive Earth gave rise to the chemicals, the precursor molecules that would lead to cellular life, ultimately, because the conditions were conducive to that. And in fact, through the modifications that he would make over the course of his life, he was able to demonstrate that you could make a wider range of the simple organic molecules that could be assembled into larger organic molecules that would form the basis of cellular life. We now understand that the Earth's early atmosphere was not a combination of these gases. It was a little bit more complex than this. Some of these gases were present, but there were other gases that were present as well, including carbon dioxide, carbon monoxide, sulfur dioxide, perhaps some nitrates or nitrites. And when you start modifying the atmosphere to resemble our modern understanding, our modern models, you don't limit the number of molecules you can make. You actually increase the number of different types of molecules and the variety of molecules that you can make in a device like this. While this doesn't demonstrate that the molecules that would lead to life were actually synthesized on Earth, there are other models that people have proposed for the origin of those molecules. It does indicate that if this chemistry did occur on the Earth, it certainly could give rise to the precursor molecules. So irrespective of whether the molecules were produced via the chemistry that existed pre-cellular life on Earth, or whether they came from somewhere else out in the universe, because that's another hypothesis, we do know that those molecules eventually turned up on Earth and did assemble into the larger molecules that would eventually lead to cellular life, because life is here. It's as simple as that. Life is here. So the chemistry, wherever it occurred, is possible. And certainly if the conditions that have been proposed and our latest models have a lot of evidence that support them, then those conditions certainly favored the chemistry that would give rise to the organic molecules that would eventually lead to cellular life. If you want to learn more about that, and if you're intending to get a biology degree, you will actually learn more about that when you take our General Biology 2 class, because we talk a lot more about the various things that would have happened pre-life on Earth that would have led to cellular life, because there are a number of other steps other than making the simple precursor molecules. But nonetheless, we do know that the chemistry is most definitely possible, and precursor molecules most definitely can be created abiotically under conditions that are thought to have existed based on our current models on the early Earth. And as I said, our models have a pretty good body of evidence to support them. So all of this comes out of Waller's simple observation that you can make urea by mixing chemicals together in a beaker. Not only does he give rise to our industrial chemistry, but he also gives rise to our further understanding of the origin of life here on our planet in the form of an abiotic chemistry that can give rise to the organic molecules that are found in cells. So, first break and our first concept check. So, if you want to take a bathroom break, if you want to go get a drink, grab a snack, now's the time to do it. Put us on pause. Remember, it doesn't matter if you put me on pause, doesn't matter if you turn me off for a while to go think about things or put me on pause to think through the questions. I'll still be here when you come back. So here are the questions for your consideration at this point. Why would the appearance of urea crystals have been so surprising to Wohler? And if Miller's apparatus did not contain electrical discharges, we know that organic molecules are not generated. In fact, Miller did actually do that experiment. He turned off the electric discharges and there were no organic molecules created. Why? I'll be back in a few seconds. Okay, welcome back. Whether you pause to go and take a break, or whether you pause to think through the questions, or whether you left for a while and came back to pick up where you left off. We're back. So let's take a look at the questions. Why would the appearance of urea crystals have been so surprising to Wohler? For two reasons. Number one, he did not expect urea to be synthesized in the absence of life. Remember, vitalism, the philosophy that ruled at the time, said that organic molecules could only be made by organisms, and he knew darn well there was nothing of organism origin in those beaker, in the beaker where he found his urea crystals. And number two, he would not have expected mixing inorganic chemicals together to give organic molecules, because no one had ever done it before. This was the first time this had happened. And because he couldn't remember not having anything organic in there, because he couldn't remember precisely the history of that beaker, it was very surprising to him to find urea in the beaker. It was something totally unexpected that lay totally outside of everything that anybody would have predicted under the circumstances, given the knowledge at the time. So it must have come as quite a shock to Wohler to find urea crystals in the beaker. In the absence of electrical discharges, Miller's apparatus does not generate organic molecules. Why? Well, the reason is because it requires energy to build molecules. And this is a building process. When you want to create molecules, when you want to carry out chemical reactions, you need to supply energy. And the energy in this case, in Miller's device, is provided by the electrical sparks. It's electrical energy, heat energy and electrical energy released by the electrical sparks. And so when you turn the spark apparatus off, you take away the energy necessary to drive these reactions. They're not spontaneous. They do require a source of energy. And so the electrical energy, or the heat energy and the electrical energy from the sparks, are what are providing the energy to drive these reactions. Take the energy away, the reactions don't occur. So that's why Miller's device can't make organic molecules in the absence of an electrical spark. I'm not sure what the heading says here. Let me drop this down. Formation of bonds with carbon. Okay, so the next thing is to think in terms of what happens when carbon starts to form covalent bonds. It's not just that carbon forms 4 covalent bonds, although that's important. It's what happens when you do form those 4 covalent bonds. So we need to remember, and here's our old friend methane again, we need to remember that the 4 covalent bonds are formed by these 4 hybrid orbitals that point to the points of the tetrahedron. So when we start linking carbons together to form chains, what we're really doing is linking together these tetrahedrons. And so we are linking together 3-dimensional shapes, which means that even though we draw diagrams of the molecules in 2 dimensions on a sheet of paper, we do need to start thinking and visualizing in 3 dimensions, because that's how we start to realize the huge flexibility that comes about by linking carbons together in chains, by linking these tetrahedrons together. So when we start doing that, we start seeing that the tetrahedrons form 3-dimensional shapes. So here's our old friend methane again, and you can see that methane forms that tetrahedral shape. So here we are now linking together 2 tetrahedrons to form the next member of the class of organic molecules that methane is the first member of, called alkanes. So let me put the name in here. Oops, remember to turn the pen on, I guess. So alkane. And notice that we have the suffix or ending ane. As soon as you see ane, that tells you that you've got carbons in the chain linked together by single covalent bonds. So when we look at the second member of the alkane group, ethane, the ETH is the prefix that tells us that it's 2 carbons. The ANE is the suffix that tells us that the 2 carbons are linked together by a single covalent bond. So this is ethane. And ethane is 2 tetrahedrons, 2 carbon tetrahedrons linked together. And so you can see that one point of the tetrahedron for each carbon forms the covalent bond between the 2 carbons, and then the other 3 molecular orbitals that form the rest of the tetrahedron are linked to the hydrogens that point out in 3-dimensional space. Now, I don't have a model of ethane because I have used all of my carbons from my molecular model kits to build molecules that are important for other parts of the chapter. So I don't have a model of ethane. I think I'm going to have to buy another kit so I can build a few more models. But I can kind of show you this. Let me just take apart one of my models for a minute so I can show you this. So here's the 3-dimensional model of ethane. So if you can't really see it in the diagram, here is the ethane model, and I'll try and get my fingers out of the way so that you can see it. And so you can see here's one tetrahedron. Here's the other tetrahedron. And then the 2 tetrahedrons, points of the tetrahedron come together, and here's the base of the tetrahedron with the hydrogens on it. So it starts to give you a 3-dimensional model, even though we would draw this. Let's see what I've got here. Put my models back together so I can show you the things that I want to show you later in the chapter. So we might draw ethane if we were drawing it out. Well, you can see it's drawn out. I don't need to, it's drawn out here. So you can see how we draw it in 2 dimensions, but we have to remember that we're dealing with 3 dimensions. So there's a whole family of these alkanes, and there are prefixes. You know some of them. So you've heard of propane, so prop is for 3. You've heard of butane, but is for 4. 5 is pentane, 6 is hexane, 7 is septane, S-E-P-T. 8 is octane, 9 is nonane, 10 is decane. And so the prefix tells you how many carbons. The ANE tells you that it's a chain of carbons with the carbons linked by single covalent bonds. We can, however, form double bonds between carbons. And I can model this for you using this model. We're going to see this again for something else a little bit later. But here you can see the double covalent bond between the two carbons, and then you can see the remaining covalent bonds sticking off from the side. And this now forms not a three-dimensional model, it forms a more planar model because of the presence of the double covalent bond. And that was the point of showing this. It's a very planar type of molecule rather than a more three-dimensional molecule in the case of ethane. But notice too that the name of the molecule has changed. We still have the ETH because it's still two carbons linked together, but now we've got a new suffix, ENE, which tells us that there is a double bond in the molecule, and this forms the beginning of a family called alkenes, where the ENE says there's a double bond in there. And the early alkenes come in a single form because there's only one way of putting a double bond in a chain of two carbons. There's really only one way of putting a double bond in a chain of three carbons. When you get to a chain of four carbons, then things change because you can form two different molecules depending on where you put the double bond. The same is true when you link atoms together by single covalent bonds or carbons together by single covalent bonds. There's only one way to link two carbons together. There's really only one way to link three carbons together. But once you link four carbons together, there are two alternative ways of doing that, which starts us down the road of thinking in terms of the diversity of shapes of molecules that carbon can form. Because right now we're still thinking in terms of what we refer to as straight chain molecules, where the carbons are linked together in a daisy chain, one behind the other. But that's not the limit to what carbon can do. So we'll come back and we'll talk about forming covalent bonds with hydrogen, oxygen, nitrogen, and carbon in more detail towards the end of the chapter when we start looking at these in combination. But just remember that all of these atoms, which represent the big four as far as biology is concerned, all of them form covalent bonds. They can form covalent bonds with themselves, they can form covalent bonds with each other, and they can, in the case of hydrogen, oxygen, and nitrogen, also form covalent bonds with carbon. So once we start to build the carbon chains, we also need to remember that we can form additional covalent bonds out to nitrogen and oxygen and hydrogen from that carbon chain, or to combinations, different combinations of hydrogen, oxygen, and nitrogen. Everybody forms covalent bonds. But because of their valence, the number of electrons required to fill out the valence shell, they can form different numbers of covalent bonds. So hydrogen with a valence of 1 forms 1 covalent bond. Oxygen with a valence of 2 covalent bonds. Maybe I should put that in here. That's not particularly neat. And of course, carbon, 4 covalent bonds. So the valence in the case of these atoms tells us how many covalent bonds they can form. And then, of course, we also need to remember that oxygen, we haven't really emphasized this very much, but also nitrogen tend to form polar covalent bonds. Now, it does depend a lot on what they're forming their covalent bonds with. So if oxygen forms a covalent bond with itself, it's going to be a non-polar covalent bond. But if it's forming covalent bonds with hydrogen or carbon, they're going to be polar. Nitrogen, likewise, with hydrogen at least, can form polar covalent bonds, and to some degree with carbon. Carbon and hydrogen tend to form non-polar covalent bonds with each other because although they may be slightly different in electronegativity, and remember electronegativity is what determines the nature of the covalent bond, they are so close in terms of electronegativity that the bonds are, for all intents and purposes, non-polar. So that's what we need to remember as well. So we'll talk a lot more about this towards the end of the chapter when we talk about the diversity of, or we start to talk in terms of the diversity of organic molecules. We won't talk in detail, that's for the next chapter, but we'll be beginning that discussion at the end of this chapter. So now that we understand that carbon can start to form these chains, we now need to start to think in terms of the diversity of chains that can be formed. So here is ethane, here is propane. Once we get to butane, so you can see what I was talking about, there's only one way to link two carbons together in an alkane. There's only one way to link three carbons together because the chain is the chain. But when it comes to four carbons, there are two ways of linking four carbons together. One is the straight chain. So this is the, let's make this a little neater. This is referred to as a linear chain, and this is referred to as a branched chain. And so I can show you these two because I do actually have models of these. So here's the linear version, and you can see immediately that the two-dimensional model or the two-dimensional diagram on paper does not really do justice to the molecule. In terms of its three dimensions, it looks very different. So even though we draw the molecule with the carbons running in a straight chain from left to right or right to left, however you want to look at it, in fact, we have to remember that we're linking those tetrahedrons together, and we're doing it in we're linking three-dimensional structures, the tetrahedrons together. So this is considered to be a linear chain or straight chain, but you can see that it has more of a herringbone pattern in true three dimensions. That's why I like the models, because it gives you a better feel for what the molecules actually look like. So that's the linear version. Here's the branched version. So here's our three carbon chain, and then here's the branch off the side. And the reason why it's important to recognize this is that while these both have the same chemical formula, there are 4 carbons and there are 10 hydrogens on each of these molecules. And just to prove it, let's add them up. 1, 2, 3, 4, 4 carbons. 3, 5, 7, 10. So there's 10 hydrogens on that one. So it's C4H10. When we look at this one, 3, 6, 9, plus 1 here, that's 10. So both of these are C4H10. But the one thing that you can say about these two is that while they may have the same chemical formula, C4H10, they are definitely not the same molecule. And so there are a couple of things that grow out of this. One that we'll talk about right now, and one that we'll talk about in a little bit more detail in a few moments. So the first thing is that when it comes to identifying the molecules, we'd like to have names on them. And so when it comes to ethane and propane as alkanes, there's no confusion here because there's only one way of linking these carbons together. So you have ethane, linear molecule. You have propane, linear molecule. When it comes to 4 carbons, you still do have butane, linear molecule. But when we get to the branched version with 4 carbons, we can't really say that it is butane anymore because it's not 4 carbons in a linear chain. So one of the ways we differentiate this is to say that the butane here in its linear form is N. Not doing this very neatly right now, so let's try this again. This is N butane. So when we think in terms of the straight chain molecule, we say N butane. When we think in terms of the linear molecule, the branch molecule, one name is isobutane because these two represent what are referred to as isomers. And that's what we'll talk about in a few minutes. But in terms of giving it its proper chemical name, because there are rules that govern the way we name these molecules, and they're kept by an organization called IUPAC, which is the International Union of Physical and Applied Chemistry, IUPAC, and they are the rule keepers for naming conventions. And so in the case of the naming convention for these branch molecules, the first thing that you have to do is decide what is the longest linear chain of carbons, and the longest linear chain that we can find here is 3 carbons long. Well, a 3-carbon chain is a propane. So at the heart of the name is the propane, and you can see it there. So let me underline that in red to highlight it. So that propane reflects the 3-carbon chain. The branch is attached to the middle carbon, and when we have chains of carbons, we like to number the carbons, and you'll see that that becomes even more important as we dig deeper into the chemistry of cells later on in the semester. And the rule in applying numbers to a carbon chain is to number the carbons in such a way that the unique feature of the molecule is attached to the lowest number carbon possible. Well, in a 3-carbon chain with the branch attached to the middle carbon, it doesn't matter which direction you number the carbons in, from left to right 1, 2, 3, or from right to left 1, 2, 3. The middle carbon is always number 2. So we say that the branch is attached to the number 2 carbon. So that's why the 2 is here. And that reflects that the branch is attached to the number 2 carbon. Then we look at the nature of the branch. The branch looks very much like methane. So here's methane again, and you'll recall that methane is a carbon with 4 hydrogens. The big difference between the group attached, the branched group, and methane is that one of the hydrogen is missing, but that missing hydrogen accounts for the covalent bond, or it's necessary to make the covalent bond to the carbon chain. So this is not called methane, it's called methyl. So there's the methyl part of the name, and that reflects this. So once you look at the name 2-methyl-propane, it tells you what the molecule is like. The propane tells you that it's a 3-carbon chain at the base of the molecule. The 2 tells you that there is a branch, or there is something attached to the 2nd carbon, and the methyl tells you exactly what it is that's attached to the number 2 carbon. And if you know the naming conventions, the naming rules that are maintained by IUPAC, which are universal, everybody uses them, then even the most horrendous name of an organic molecule can be deciphered, and you can draw out a diagram of what the molecule looks like. That's why the rules exist, because chemistry, like all of science, is a universal process. And while everyone may not be able to communicate to each other conveniently in the same language, everyone understands the language and the naming rules of the diagrams. So even if you can't easily explain yourself in a common language, you can draw a diagram. you can provide a name and everybody knows what the molecule looks like. That's the universality of chemistry. Now, I mentioned that you could put double bonds in there, and I showed you a molecule that had a double bond, and we'll talk about this one in more detail in a moment. But again, if you put a double bond in a chain that has 2 carbons or 3 carbons, it doesn't matter where the double bond is, the molecule is exactly the same. So if you put the double bond in here, it doesn't matter. It's exactly the same. If we put the double bond in here, that's no different than putting the double bond here. We just simply flip the molecule around. So the numbering would still say number from the most unique part of the molecule. It really doesn't matter, because whether we've got it here or here, we just flip the molecule around. Nothing matters, nothing changes. But when it comes to a 4-carbon chain, now it matters, because we can put the double bond in here, or we could put it here, but that's just flipping the molecule around. Or we can put it between the middle 2 carbons. And this is very different from this. These are not the same molecule. So we now have to think in terms of how do we name this molecule, these 2 molecules, to reflect what we're looking at. Again, the naming conventions say we number the carbons, and we again number the carbons in such a way that the unique feature is attached to the lowest carbon possible. So we could number the carbons in this molecule 1, 2, 3, 4, and well, 1, 2, 3, 4, or we could go 1, 2, 3, 4. But remember, lowest numbered carbon possible. So if we number 1, 2, it means that the double bond is between carbons number 1 and 2, rather than numbers 3 and 4 if we put it down here, and or we've numbered from 1, 2, 3, 4 in the opposite direction. So we number 1, 2, 3, 4, and we call this 1-butene, indicating that the double bond starts at carbon number 1 and connects to carbon number 2. In this case, it really doesn't matter whether we go 1, 2, 3, 4, or 1, 2, 3, 4. It's between carbons 2 and 3, but we call this 2-butene to indicate that the double bond is between carbon number 2 linking to carbon number 3. So again, the naming rules tell us something about the molecule, and we can decipher what the molecule looks like by understanding the naming rules. So now we've got linear chains, we've got branch chains, we've got chains with double bonds between the carbons. We're not done yet, because carbon can now form ring structures. And I don't have a model of cyclohexane, because I don't have enough carbons in my modeling kits. But you can see that now the chain folds around on itself and links back up again. So this chain, this ring contains 6 carbons. That makes it a hex something. The bonds between the carbons are all single covalent bonds, so that means it's an alkane, so it becomes a hexane. But because it's circular, it's called cyclohexane to separate it from N-hexane, which would be the linear form of the hexane. And then we have a very unusual molecule called benzene. And the name benzene doesn't actually follow the IUPAC naming conventions. But the reason why is because this name was given to this molecule very early on in the history of organic chemistry. And it is such an important molecule in industrial chemistry that it forms the basis of a whole area of organic chemistry by itself. The nature of the benzene molecule and molecules that are built out of benzene. It's a very important starting point for a lot of organic chemistry. And because of that, rather than try and create a IUPAC standard name for this molecule, we use the traditional name benzene because it's so important to our understanding of organic chemistry. So we hold on to that name. So now we can understand why carbon starts to take on such an importance. It's not just that carbon can form 4 covalent bonds with itself or with other atoms. It's that even just in thinking about carbon forming covalent bonds with itself, there is such a diverse array of different types of molecules and structures of molecules that carbon can form. And we haven't even begun to think about adding anything else to the carbon chain other than hydrogen at this point. So all of these molecules are also examples of hydrocarbons. Carbon chains with hydrogen attached to the covalent bonds that are not involved in linking the carbons together. So basically a carbon chain with just hydrogen attached to it. That would be a hydrocarbon. And this is the basis for all organic molecules. These are all organic molecules. Some occur naturally in nature. Many are synthetic. Breakdown products of working with oil and gas and the like. That can be derived from that, derived from coal, all sorts of things that we can make as part of our industrial chemistry. So these are all molecules that we can make, but they're all examples of hydrocarbons. We're going to see how we can modify that basic hydrocarbon before we're done this morning. Before we look at how we can modify it, let's go on and look at another concept that comes out of this. And this is the idea of isomer. Now, I used the term isomer earlier when we started talking about butane or N-butane and isobutane. So the linear version of butane and the branched version of butane, or to give it its proper name, 2-methyl propane. They are an example of one type of isomer, the so-called structural isomers. So here's another example of a structural isomer, but let's just take a look at the two that we're already familiar with. So here's the linear version of butane, and here is the isobutane or 2-methyl propane. So again, we have a common chemical formula, C4H10, 4 carbons, 10 hydrogens, which we demonstrated when we first looked at these molecules. But these are arranged into 2 different structures. And that's why these are referred to as structural isomers. Because while they share the same chemical formula, the arrangement of the atoms are very different. They're arranged into very different structures, and as a consequence, they have very different chemical properties. So a basic definition of isomer, and I'll put this on the previous slide. Actually, it's here on the previous slide, so let me just highlight that. So in order to remember what an isomer is, molecules with the same molecular formula, but different structures and different properties, or different arrangement because, as you'll see, structure is somewhat in the eye of the beholder. So these structural isomers clearly do have different structures. There's no denying that these are 2 very different structures. When we look at some of the other isomers, you're going to say they don't look that much different in terms of structure, so why are they not referred to as structural isomers also? Well, as you'll see, there are very much differences in the arrangement of the atoms, even though structurally they may look very similar. So structural isomers very much so have the atoms arranged into very different structures, even though they share the same chemical formula. So the next type of isomer, indicated here as the cis-trans isomer, are also known as geometric isomers. So AKA so the book has got into the habit of using the term cis-trans isomer chemically, and you would learn this in an organic chemistry class, which some of you may very well be taking in the future if you're getting a biology degree. You will have to take organic chemistry. You'll learn them as geometric isomers because that's the chemical term that we use for them. And the key feature here, and this is really important to recognize, the key feature is the presence of the double bond between the two carbons. And that's what's really critical here, because you're going to see other molecules. You'll see some this morning, or you'll see some in this presentation, where there's a double bond present in the molecule, but it's not between two carbons. And that means you can't have these geometric isomers or the cis-trans isomers. The only way that you can have cis-trans isomers or geometric isomers is if there is a double bond between two of the carbons in the chain. So here are the two examples. So this is the first form. This is called the cis isomer. So you can see the double bond in here. And then we've got two white balls, which are typically considered to be hydrogens, and then two orange, which is something else. And the reason why this is called cis is because cis is the Latin prefix for same. And so these are same side isomers. These two white balls are on the same side. The two orange balls are on the same side of the molecule. And the reason why this is important is because of what the double bond does. But before I show you what the double bond does, let me show you the other form. So here's the other form called trans. And trans is the Latin prefix for opposite. So here, the two whites are on the opposite side of the double bond, and the two oranges are on the opposite side of the double bond. And so this is the trans form. So here's the cis form, and here's the trans form. And why it is important to recognize that these exist because of the double bond between the two carbons is because the double bond creates an inflexible molecule. You can't twist this. It can't rotate. The only way that you can go from one form to the other form is to literally break one of the two covalent bonds between the carbons, rotate everything around, and then reform the double bond again. But breaking and forming double bonds, remember, is a chemical reaction. That requires energy. You cannot spontaneously change this form to this form, or vice versa. That can only be done by a chemical reaction. So these are two very distinctly different molecules. And when we talk about the macromolecules, the large molecules in cells in the next presentation, this is going to become very important in one of those macromolecules, the fats. And I'm not going to tell you what the key is there, but think about what you've heard about fats, particularly over the last 10 years or so. And you might start to remember that trans sounds familiar when it comes to thinking about fats. And obviously it's got something to do with these types of isomers, but more to come in the next presentation on that one. The last type of isomer is the hardest one to try and figure out. It's got a fancy name called enantiomer. Another way of saying this is mirror image. And the diagram here is tried to illustrate now an idea that we often use in trying to explain this. So if you take your two hands and you hold them up like this, if you slide them sideways, just like that, nothing really matches up on your two hands. They're out of sync with each other. But if you separate them back like this again, put them thumb to thumb and rotate on the tip of your thumb, each finger aligns with its partner on the other hand, and they perfectly line up. And the reason why is because your left hand and right hand are mirror images of each other. If you hold your right hand up to a mirror, as I'm holding this up to the camera on my computer, when I look at that image, it actually looks like my left hand. If I hold my left hand up away from the camera so that I'm looking at it away from the camera, it looks exactly like the image of my right hand in the camera because they're mirror images of each other. And that's the nature of these isomers. So I'm going to show you my models of the two isomers because I've got two models of these, of the two forms of these enantiomer, and I'll show them to you separately, and then I'll show them to you side by side. And I'm going to hold them in exactly the same fashion. So I'm holding this one by the orange ball. You can see that under my fingertips. I've got the purple. Well, to me, it looks purple. In the camera, it looks like it might be blue, but it's actually whatever color it is. There's a brown one at the back, and there's a white one over here, okay? So here comes the other one, and I'm going to hold it in the same orientation, but now you can see that the brown and the white have swapped sides. Orange is still at the top. Blue or purple is still facing you, it's still facing the camera, but the brown and the white have swapped sides. So here they are side by side, and you can see that what I'm talking about here. So if I slide them like this, just as I did with my hands, nothing matches up. But if I put the whites together and rotate them around on the whites, just like I did my hands with my thumbs, everything lines up with the atom on the opposing molecule, just as my hands did, because they're mirror images of each other. And there is a key feature of these molecules that allows you to recognize that they're possible. And it has to do with the central carbon. This is called an asymmetric or chiral carbon. And the reason why is because it has 4 different And the reason why it's considered to be asymmetric is because it has 4 different things attached to that central carbon via the 4 covalent bonds. So I've said 4 different units, and the reason why is because when we look at bigger molecules and we look for chiral carbons or asymmetric carbons, we have to recognize that it may not be single atoms that are representing the different things that are attached to the central carbon. It could be combinations of atoms. But if we look at this central carbon, it's got a white, a purple, an orange, and a brown attached to it. There's your 4 different units attached to the central carbon. This one does as well, but the arrangement of the 4 different things is different on the two molecules. Hence, the possibility of mirror image molecules or enantiomers. We used to use the designation L and D, L for left-handed, D for right-handed. It has to do with the interaction of these molecules with a form of light called polarized light. That's something that you might learn about if you take organic chemistry, but we don't need to go into the details anymore in this presentation as far as that is concerned. They have changed the designation recently, and they now use the letters R and S to designate the 2 different enantiomers, but it's basically the same idea. It's just a new naming system or nomenclature for designating the 2 mirror image forms. But just think in terms of the idea that these are mirror image molecules because there are 4 different things attached to the central carbon. So biologically and pharmaceutically, these have consequences. So in the case of amino acids, life makes and preferentially uses the L version, the left-handed versions of amino acids. And that's important because it's a product of cellular chemistry. If we do the chemistry in the laboratory, we don't make one of the 2 enantiomers. We make both of them together, what is referred to as a racemized mix. And you don't need to understand the term racemized or even remember the term racemized, but it means that there are equal amounts of the left-handed and right-handed enantiomer. And that becomes problematic because if there are drugs that come as enantiomers, we can't preferentially make one of the 2 enantiomers. We have to make both of them. And it turns out that in some cases, one enantiomer may be a useful drug, the other may be very, very harmful. And we learnt this the hard way back in the late 50s and early 60s with a drug called thalidomide that was used as a sedative for pregnant women who were suffering from morning sickness, which they usually suffer from during the first trimester, the first 12 weeks of pregnancy. And the reason why that is a problem is that at that point in development, the embryo is very, very sensitive to disruption in normal development by environmental things like chemicals. And in the case of one of the 2 enantiomers of thalidomide, this was the case. One of the 2 enantiomers was the sedative. The other enantiomer was a teratogen, that is a drug that causes, or a chemical that causes birth defects. And the particular birth defects involved arms and legs so that they didn't develop properly. And there was a whole generation of kids born in Britain, in Australia, in New Zealand, and I believe also in South Africa. Fortunately, not here in America. And these are known as the thalidomide kids because unfortunately, their mothers took thalidomide during that first trimester as a sedative for morning sickness. And when kids started to be born with this birth defect, it took a number of years of careful research to identify thalidomide as being the cause. And eventually, once it was identified as a cause, it was quickly taken off the market, as you might expect. But the damage was already done. But the point behind this was that the drug company didn't realize that this was going to be a problem because no one knew that this would be a problem. No one knew that it was possible for one enantiomer to be a useful drug and the other to be really dangerous. But thalidomide taught us that that's not the case anymore. That we really do have to think in terms of how safe are both enantiomers, not just the one that we want to use as our drug of choice. We really do need to consider both of them. And so in the laboratory situation, it is possible, it's very tedious and not easy to do, but it is possible to separate the 2 enantiomers from each other so that you can test them separately. But that's not possible to do in an industrial situation where you're trying to produce large quantities. And so at the clinical testing stage, where you know that you've got these 2 enantiomers, it's really critical that the 2 be tested separately to make sure, particularly in the early testing when they use animal models or cell models to test the drugs, to make sure that both enantiomers are safe. That even if only one enantiomer is the active drug, that the other one is biologically inert so that it doesn't cause any damage. So in the case of dopamine, which is used for the treatment of Parkinson's disease, one of the enantiomers is a naturally occurring form of dopamine that's made in a set of cells found in the brain. When those cells stop making dopamine, we have the beginnings of Parkinson's disease. So you can treat it by supplying a synthetic form of dopamine, but the synthetic form has both enantiomers present. Fortunately, the non-biologically active form of dopamine is biologically inert. So it is possible to give synthetic dopamine to offset the loss of dopamine from Parkinson's disease, at least for a while, because it turns out that eventually the synthetic dopamine has no effect. It becomes less and less effective until finally it has no effect at all, or it has a very, very short-term effect, minutes. And you just can't keep giving individuals constant supplies of dopamine because eventually it becomes ineffective, totally ineffective. And this is the condition that, oh, I've forgotten his name now. Michael J. Fox. You would know him from the Back to the Future films. He was the lead in the Back to the Future films. He has, unfortunately, a very early onset form of Parkinson's disease. Normally, it's a disease that affects people in their 60s and 70s. He started to develop symptoms in his late 20s and early 30s. So he has an early onset form. And he was treated with dopamine for a number of years so that he could continue acting without showing signs of the disease. But eventually, the dopamine became so ineffective that he decided to continue acting and let his symptoms be visible. And so there were situations where he was on talk shows and a couple of shows that he was that he acted in, where he did not even try to disguise his Parkinson's disease because there was no way that he could. But for a while there, he tried to disguise the symptoms using dopamine, but eventually it got to the point where he couldn't disguise it or mask it, let's put it that way. Because he's been pretty open about the fact that he has Parkinson's disease, and he's been very vocal in advocating for research on Parkinson's disease. So that was one of the things that brought dopamine and Parkinson's disease more to the forefront of public awareness. Some other drugs that are also found as enantiomers: ibuprofen. So if you take ibuprofen as a painkiller, the S form, remember I said that we now use R and S rather than L and D. So if you take ibuprofen, the S form is the active painkiller. The R form is, fortunately, biologically inert. So you take a pill that contains both enantiomers. This one does nothing. It's biologically inert. This is the one that acts as the painkiller, and it works quite well. If you have asthma and you carry a rescue inhaler, it contains the chemical albuterol. And albuterol also comes as two enantiomers, R and S. This time it's the R form that is the anti-inflammatory that relaxes the muscles around the throat so that you can breathe. And the S form is the biologically inert. It does nothing. So your inhaler actually contains both forms of albuterol. The R is the one that gives you the relief. The S is just along for the ride because we can't separate them out conveniently at the industrial level. And fortunately, it is biologically inactive. It's biologically inert. So these are the isomers. Three different types of isomers: structural isomers, where it's the same chemical formula, but the atoms are arranged into two very different structures, leading to very different chemical properties. The geometric isomers, otherwise also known as cis-trans isomers, where the structures look very similar because of a double bond between two carbons, but the location of the atoms is different. So in the cis form, the atoms are on the same side of the bond. In the trans form, they're on opposite sides. And then we have the mirror image isomers, otherwise known as enantiomers, left-handed and right-handed versions, and they exist because there are 4 different things arranged around the central carbon, which causes that carbon to be asymmetric, or to use the more appropriate chemical term, it is a chiral carbon. So let's take another break and let's put your information to use by asking you to look at these diagrams of molecules that we were talking about previously and to ask a simple question. Which of these pairs of molecules are isomers, and what type of isomers are they? So if you want to go and take a bathroom break, grab a drink, get a snack, put me on pause, go do that, come back, think about the question, and I'll come back and see you with the answers in a moment. Okay, so let's take a look. We can immediately rule out 2 sets of molecules. The ethane-propane pair. Oops. I need to select the pen again. So we can rule out the ethane-propane pair. Why? Because remember our definition of isomers says same chemical formula but different structures. These don't have the same chemical formula. This has got 2 carbons, this has got 3 carbons. So this would be C2H6, this is C2H8. Very different chemical formulas, so they can't be isomers. Likewise, cyclohexane and benzene, while they look very similar, they're both ring structures, the number of hydrogens is very different between cyclohexane and benzene. So again, similar structures, different number of atoms, different chemical formulas. So again, they can't be isomers. But with 1 butene and 2 butene, 4 carbons, and 1, 2, 3, 4, 5, 6, 7, 8 hydrogens. So this would be C4H8. 1, 2, 3, 4, 5, 6, 7, 8. Also C4H8. But different arrangements of carbons and hydrogens. So are these potential isomers? Yes, same chemical formula, different structure. So they're potentially isomers. What type of isomers then? Well, the key is same formula, different structure. So these would be structural isomers. Then of course down here, well, these are the original structural isomers that we looked at. So again, these would be structural. So yes, there are 2 sets of isomers here. Both of them are structural. Hopefully you saw that. So now we come to the last section of this chapter. Everything that we've talked about up to this point has involved carbon skeletons with hydrogens attached. In other words, hydrocarbons. I pointed that out to you before. The problem with hydrocarbons, and this is seen with one set of hydrocarbons that are found in cells, and we'll talk more about these next week, the fats. The problem with hydrocarbons is they don't dissolve in water. And that is a real problem because, as you may recall from the last presentation, when we talked about water, one of the key features of cellular chemistry is that in order to participate, you've got to be hydrophilic, soluble in water. If you are a hydrocarbon, you are hydrophobic and therefore not soluble in water. And most of the organic molecules that are found in cells start out with a hydrocarbon framework. In other words, they are potentially hydrophobic and therefore not soluble in water at all, which would mean that they wouldn't participate in cellular chemistry, and that would be a huge problem. Now, the fact that fats are hydrophobic and don't dissolve in water actually is not a problem, because when we look at the role of fats in cells, which we'll start to do again next week, you'll realize that the insolubility of fats in water actually is a property that we need because of their role in cells. But for the other large molecules that are found in cells, the carbohydrates, the proteins, and the nucleic acids, DNA and RNA and so forth, those do need to be soluble in water. They're going to be in a water-based environment, a water-based solution, and they're going to be involved in chemistry that involves being dissolved in water first. So how can we take a basic hydrocarbon structure and turn it into something that can dissolve in water? Well, that's where a group of molecular structures form from mostly oxygen, hydrogen, and nitrogen can change the nature of the basic hydrocarbon skeleton. And we refer to these as functional groups. And there are 7 functional groups in total that are going to become important to us throughout this semester. So here are the 7 functional groups: the hydroxyl group, the carbonyl group, the carboxyl group, the amino group, the sulfhydryl group, the phosphate group, and the methyl group. 6 of these are polar, meaning they are themselves soluble in water. And the presence of one or more of these functional groups on a hydrocarbon chain changes that chain from being insoluble because it's a hydrocarbon to something that can be dissolved in water because it now has a hydrophilic functional group on it, something that can dissolve in water. So having these functional groups, and particularly those that are polar, turns out to be really, really important. Now, there is a diagram in the textbook, and I've reproduced that in the PowerPoint slides, and this is part of that diagram, that not only shows you what the molecule looks like or the functional group looks like as a separate unit, but also shows you the functional group included in molecules. And it's really important that you learn to be able to recognize the functional groups in diagrams, both separately, like this, and also as part of molecules. Every semester when students come back to talk over their exams and examine the exam questions and try and figure out why they got exam questions wrong, which you are able to do during office hours. I do not release the questions publicly because I want to maintain the integrity of those questions, because I worked really hard to create my exam questions and I want to be able to reuse them whenever I choose to. So I want to make sure that I retain as much integrity as I can for those questions. So if you want to look over your exam questions, you're welcome to do so after the exam deadline has passed and the exams have been graded. You can come to office hours and you're more than welcome to sit down with me and go over the exam, and we can talk about why you got questions wrong. And based on my experience, one of the things that is going to create a headache for you is being able to recognize functional groups, being able to recognize molecules in diagrams. So I want to give you a heads up right now. It is very, 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 very important that you look at these diagrams and you be able to recognize the different types of functional groups and the molecules that they exist in. If you want a suggestion on how you might do this, I always found flashcards to be very, very useful here. On one side, I might have the functional group. On the other side, I might have its name. On one side, I might have a functional group and I might have its properties on the other side. On one side, I might have a functional group and a molecule that contains it on the other side. Or the name of a functional group and a molecule that contains it on the other side. And so you ought to be able to go backwards and forwards, and you may need to have 3 or 4 flashcards for each of the functional groups in order to be able to quiz yourself on all of the various chemical properties and structural features that you need to know about each of these functional groups. But it is really, 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 really important, I cannot stress this enough, that you be able to recognize each of the functional groups separately in a diagram and in a molecule in a diagram. You must also know their chemical properties. So I'm going to go over some of those chemical properties with you. I'm trying to think how I might do this. So I'm going to pull this down to the bottom for a moment. I don't need to see the text at the bottom as it does the transcript, so that I can look at the functional groups at the top of the table and maybe make a couple of notes as we go. Let me just check the color that I've got in here. I'm in black, so that's what I want. Okay, so the hydroxyl group, you can see what it looks like there. It's an OH group. So the hydrogen and the oxygen are covalently bound to each other through a single covalent bond, and then the second covalent bond of the oxygen allows it to covalently bind to a carbon chain. You can see that here in ethanol, which is the type of alcohol that is found in wine and beer and spirits. And there you can see the hydroxyl group covalently bound to the carbon chain. This is polar. So it makes the carbon chain soluble just by having the presence of the hydroxyl group on the carbon chain. This is why alcohols in general are soluble in water. This is also found in sugars. So sugars typically have lots of hydroxyl groups on them as well. It's the reason why sugars dissolve in water very, very easily. The next one is the carbonyl group, and here you do see a double bond. Now remember, I cautioned you that you will see double bonds elsewhere other than in the geometric isomers or the cis-trans isomers. But remember, in the isomers, the double bond is between two carbons. Here, the double bond is between a carbon and an oxygen. So this cannot give you cis-trans isomers or geometric isomers. The double bond has to be between two of the carbons in the carbon chain for that to be possible. So bear that in mind. The carbonyl group is the oxygen double bonded to the carbon. It can be in one of two forms. It can be terminal, in which case it is an aldehyde. It can be within the molecule, in which case it is a ketone. So the aldehyde is terminal. It's at the end of the chain. Whereas in the ketone, it's somewhere inside the chain. It is also polar. Because of the oxygen. I mean, we know oxygen is electronegative, so it's going to, this double bond is going to be somewhat polar because of the presence of the oxygen. That means it's present on any carbon chain is going to make that carbon chain soluble as well. And this is also found in sugars. And in fact, there are two different types of sugars based on where the carbonyl group is located in the carbon chain of the sugar. So there are aldehyde sugars, because the carbonyl group is at the end of the chain. These are sometimes referred to as aldoses. And you'll get used to the fact that OSE is the suffix or the ending of the name of every sugar. So an aldose is an aldehyde sugar. And then you have ketone sugars, where the carbonyl group is attached to a carbon in the middle of the carbon chain of the sugar. And they're called ketoses. Again, OSE tells you that you're dealing with a sugar. And you're going to see it. If you've never thought about that, Just think about this: sucrose, glucose, fructose, lactose, maltose. They all end in OSE because they're all examples of sugars. Okay, so that's the carbonyl group. The next one is the carboxyl group. This one is acidic because, as you can see from this diagram, when you dissolve something like acetic acid that has this carboxyl group, the hydrogen that's attached to the oxygen in the hydroxyl group can come free because of the pulling force of all of these oxygens. And this is the acidic hydrogen. And remember, an acid we define as a substance that adds hydrogen ions to a solution or increases the amount, the concentration of hydrogen ions. So the fact that this hydrogen ion can break free from the carboxyl group when it's in solution means that this substance is any substance with this group on it can potentially act as an acid. So acetic acid, the active ingredient in vinegar, is an acid. And again, this is polar, as you might guess. So where might you find an acetic acid? Well, in combination with the next functional group called the amine group, you'll find these in amino acids. Right? Amino referring to the fact that there's an amine group, and acid referring to the fact that there's a carboxyl group. Now, when you look at the nature of the carboxyl group, you can see that it looks like it's the combination of a carbonyl group and a hydroxyl group. That's why it's called the carboxyl group, for a combination of carbonyl and hydroxyl, because that's in fact what it is. But it's a single functional group. It's the acidic functional group. So the next functional group is the amine group. This is a nitrogen with two hydrogens attached to it. The third covalent bond is what attaches it to the carbon chain. This is polar and it is basic, as the diagram suggests, because it can bind a hydrogen out of solution. And the reason why is because the nitrogen has a pair of electrons in the fourth molecular orbital, because nitrogen forms the 4 molecular orbitals just like oxygen does, just like carbon does. But because of the number of electrons in the valence shell, 3 of the molecular orbitals have a single electron, but the fourth one has 2 electrons. So you've got the fourth orbital sitting out here with a pair of electrons in it, and that attracts a hydrogen ion because you've got a big negative charge on that molecular orbital. And that hydrogen ion actually links up through something that looks like a covalent bond. It's not, it's called a coordinate bond. So, not sure why it keeps doing that. There we go. So this is called a coordinate. I'm having a lot of luck with this. Try this again. So again, while it does look like a covalent bond. It's not a covalent bond, and the reason why is because the nitrogen is supplying both of the electrons. Remember, in a covalent bond, the shared pair of electrons are shared, one from each atom involved in the covalent bond. Here, the nitrogen is supplying both of them. So while it looks very much like a covalent bond, it's not. It's referred to as a coordinate bond. But the fact that the hydrogen is being captured here from solution means that you're reducing the number of hydrogen ions. And remember, again, that is our definition of a base. A base is anything that removes hydrogen ions from the solution, and certainly the amine group does that. The next group is called the sulfhydryl group. It is weakly polar. The book typically represents this as being non-polar, but it's actually very, very weakly polar. And it is found in the amino acid cysteine, and you can see the sulfhydryl group sticking out here. Now, what is really interesting about these sulfhydryl groups is that if you have two sulfhydryl groups adjacent to each other in a molecule, you can form what is referred to as a disulfide bridge, a covalent bond between the two sulfurs. And this is a covalent bond. It's very strong, and it allows you to link these two amino acids together, which may be very important in maintaining the structure of a protein. We'll talk more about structures of proteins in the next presentation when we talk about the macromolecules, because there we're going to talk a lot more about proteins. But this is the basis for getting a permanent, because your hair is made up of a protein called keratin. And whether you have straight, wavy, or curly hair is a function of the disulfide bridges in the particular type of keratin that makes up your hair. It's genetically programmed. But if you don't like the shape of your hair, of course, you know you can go to the hair salon and get a permanent, and that will rearrange your hair into another shape. Although these days we use product that allows you to shape your hair, and then when you're done, you can wash it out and reshape it again. Permanent is just that, a permanent change to the existing hair. It doesn't reprogram the hair follicles. So the new hair that grows out is going to have the old shape, but of course you can go and get your perm done again if that's the case. And all it revolves around is using chemicals that break the existing covalent bonds, the existing disulfide bridges. You then shape the hair using curlers or whatever it is that you're trying to do. And then you add a second chemical that forms or triggers the formation of disulfide bridges, but the ones that form up are different from the original ones. You're forming new disulfide bridges that will lock the hair into its new shape. It's all about chemical manipulation of these disulfide bridges, chemical manipulation of these sulfhydryl groups. So that's something interesting. That's a sort of a sidelight to our understanding of functional groups. Let me move this back up to the top again, because now I need to be down the bottom. And let me move this one across to here so that I can see the bottom here. So the last of the polar groups is the phosphate group. And not surprisingly, this one is incredibly polar, because look at it. There are 4 oxygens, 2 of which have negative charges, which tells you they've lost something. They've given up hydrogen. So there would be an H plus and an H plus that have been given up here. So this is also acidic. And it's incredibly polar because of all of these oxygens. And so the presence of a phosphate group very much so changes the solubility of anything that's attached to it. You put a phosphate group on something, it immediately becomes very, very soluble. But as you'll see, in the very, very last closing piece of this presentation, the presence of multiple phosphate groups also has some importance. So here's an example of a sugar that has a phosphate on it. This is glycerol phosphate. Glycerol is actually a very close relative of a 3-carbon sugar called glyceraldehyde. And when you replace one of the hydrogens with the phosphate group, you actually get something called glycerol phosphate. This is very, very soluble, and we will encounter glycerol phosphate later in the semester. But this is a very, very polar, incredibly polar functional group. The one non-polar functional group is the methyl group. And the reason why we include this here is because we now recognize that the addition of methyl groups to the C nucleotide in DNA is a way of regulating genes. And so here you can see the nitrogenous base part of the C nucleotide, what actually gives the C nucleotide its C designation. And you can see the addition of the methyl group to it. So 5-methyl cytosine, the cytosine is the nitrogenous base part of the nucleotide. It's attached over here. That actually forms part of the DNA, and it's the addition of this methyl group actually, I'll change that. And say it this way. And in fact, the addition of the methyl group inactivates the genes. It's a way of turning the genes off, but we'll talk a lot more about that towards the end of the semester. So those are the functional groups. Giving you a little bit more information there. The diagram in the textbook actually contains all of this information and a little bit more in a single diagram. And if I was trying to learn this, I would be looking at that diagram and I would be making flashcards and trying to understand that diagram in detail, taking the information, putting it on flashcards and quizzing myself over and over again until I could be certain that I knew the contents of all of the flashcards with respect to each of these functional groups. It's really, really important. And we're going to build on this as we start talking about the macromolecules next week or in the next presentation, because these functional groups are what determine which of the nature of each of the macromolecules. Each type of macromolecule has its own unique collection of these functional groups, and they define the macromolecules and define the properties of those macromolecules. So again, I cannot stress enough the requirement for you to learn these functional groups as best you can. So how important are these functional groups? Well, here are two molecules, estradiol and testosterone. This is the female sex hormone. This is the male sex hormone. And you can see that the differences between the two molecules are very, very subtle, yet they have such powerful consequences, because the external gender characteristics are determined by which hormone predominates during development. And the default setting is female. So this is a pretty big deal. If you expose a developing embryo to testosterone, it will develop male external sex characteristics. If in the absence of testosterone, the embryo reverts to the default setting, which is to develop as female. And so what triggers the development of maleness is a gene that's carried on the Y chromosome. And it functions for a period of 3 days during the 6th week of development of pregnancy. And that begins the cascade that leads to the development of maleness. And the reason why this is important is because the tissue that becomes the ovaries or the testicles is the same tissue. It depends on what hormone it gets exposed to. The tissue that becomes the penis or the clitoris is the same tissue. It depends on what hormone it's exposed to. The tissue that becomes the vagina versus the scrotum is the same tissue. It depends again on what hormone it's exposed to. In the absence of testosterone, the tissues develop female characteristics. What is more important is that there are people who are functionally female. They have the gender characteristics of female. They have considered themselves to be female all of their lives, who are in fact genetically male. They have a Y chromosome. But because the cascade that leads to maleness did not take place, they were not exposed to testosterone during embryonic development. They followed the default setting and developed as a female. Unfortunately, they cannot reproduce as a functional female because they have the Y chromosome. They are sterile under those circumstances. But you could not pick them from a female who had the 2X chromosomes and developed in the normal female fashion with the presence of 2X chromosomes, because the default setting is female. And in the absence of exposure to testosterone, that's the way an individual develops. I have a picture from another biology textbook of a group of about 20 women, and they look like normal women. You could not tell them apart. You could not tell that these are individuals that have a Y chromosome because they all have a Y chromosome. This is the power of these hormones. And when someone undergoes a gender reassignment, what takes place is first removal of the tissue that gives rise to the gender hormone that that person is producing. So the ovaries and the uterus, if it's someone who is reassigning to male, and the testicles, if it's someone who is reassigning to female. And then they get injections of the appropriate gender hormone to reshape the body. And it really does reshape the body. Fat gets reassigned to different locations. Tissue develops in different ways. Hair growth patterns change. Voice patterns change, all because of the exposure to the gender hormone. These are very, very powerful molecules. And yet the differences are very, very minor in terms of the structure of the molecules. Just slight changes, slight rearrangements of the functional groups. And it changes the nature of the molecules altogether. So functional groups cannot be discounted. They can have huge consequences because of their relationship to the base carbon chain and the way they affect the behavior of the molecule as a consequence. So the final thing to look at is the relationship of phosphate groups on an ATP molecule. We're going to see a lot more about ATP when we get to chapter 8. We'll talk a lot more about ATP. But I just want to expose you to the idea that the presence of these phosphate groups is really important. And again, this reinforces the importance of functional groups. So this is the ATP molecule, adenosine triphosphate. And so it's related to an RNA nucleotide, but instead of having one phosphate group on it, as the nucleotide would have, it has 3 phosphate groups on it. And it's the close proximity of these phosphate groups to each other that represents the energy found in an adenosine triphosphate molecule, an ATP molecule. Because these 3 phosphate groups, all being extremely polar and having all of these oxygens on them, do not play nice with each other. They really don't want to be next to each other. And as a consequence, the covalent bonds that hold them together are under a lot of stress. And of course, if you're under stress, you're unstable. And what we know about this in terms of chemistry is that bonds that are under stress and that are unstable also represent a source of energy, because the two go hand in hand. If you have a lot of energy, you are inherently unstable because you're capable of changing. You can do work. You have the capacity to change. The more energy you have, the more unstable you are. So if you're unstable, you have energy. If you have energy, you're unstable. These covalent bonds, and particularly this covalent bond, and let me change the into red. This covalent bond, hold it with this oxygen and this phosphate, is incredibly unstable. But being unstable means it contains a lot of energy. So when this phosphate group is released, when this covalent bond is broken, the energy of that covalent bond is released with the phosphate group. And that phosphate group represents the energy. So when we transfer the phosphate to another molecule, the energy is also transferred. And this is actually the basis for using ATP as a source of energy in cells. So when this covalent bond is broken through a chemical reaction called a hydrolysis reaction, it involves addition of water. That phosphate group can then be transferred to another molecule, and the energy of the covalent bond is transferred with that phosphate group. And that's how the energy of an ATP molecule can be utilized by cells. This is going to become a very, very important theme as we progress through the semester, because we're going to be referring to the use of ATP in chemical reactions, in doing cellular work, whether it be mechanical or transport work, we're going to see a lot of work being driven by ATP, and this is how it's done. So this will become the subject of another discussion in chapter 8 and on into chapters 9 and 10 as well, as part of a discussion of cell respiration and photosynthesis. So you're going to see a lot more about this in the future. So one final concept, right, to close out the chapter, just to make you think about a little bit about functional groups again. So in this picture, we've got a picture of an amino acid. What does the term amino acid indicate about the structure of the molecule? And so the answer, if you remember from when we were looking at the functional groups, is that it has an amino group. And a carboxyl group or acid group. So that's what it's meant by amino acid. It's got an amino group and it's got a carboxyl or an acid group as part of the molecule. So that brings us to the end of this chapter. Stop sharing. So, been a fairly lengthy discussion of carbon and carbon molecules and functional groups, but these are all very important because we're going to build on these, particularly in the next chapter, chapter 5. But these are also going to become ongoing themes as part of our discussion of the general chemistry of cells. We'll see that more in upcoming presentations. But always, if you have any questions, concerns, or comments about anything that's been discussed in this presentation, by all means come to office hours. I'd be more than happy to help you out, help clarify anything that you're uncertain about, or to clear up any misconceptions or any other issues that you might have with the material that's been covered. So if I don't see you in office hours, I will see you next time when we start talking about macromolecules. Bye for now.